Okay, good afternoon, everybody. I know I'm the first session or the first talk after lunch, so hopefully I'm getting your attention. So yes, as the chair's mentioned, I'm Suvarna and I work for the South African National Biodiversity Institute, heading up the Citizen Science Program. So we're all quite familiar of the grasslands biome being the second largest biome in the country and most used in agriculture, mining, and so forth. But this slide tells a much deeper story. The grasslands biome is under pressure. Being the second largest biome, less than 5% is protected. Nearly 40% of the grasslands biome is transformed, yet just under 300 threatened plant taxa occurs in this biome. 150 plant taxa are actually endemic to the grasslands biome. So that's quite a grave story. And how are we here to protect our grasslands? So if we step back into South Africa and why our biodiversity is important, we're all very familiar that we're the third most biodiverse country in the world. We've got various centers of plant endemism. 6% of the world's flora occurs in South Africa. We are one of the top 10 nations globally for species richness and the second highest for plant endemism. And these are quite high stats. They will be changing in the next year, hopefully not too radically, but we are preparing for the National Biodiversity Assessment Report, which happens every 5 years. So what do we do in combating the risk of extinction of species? Over 2 decades ago, we've identified gaps and the role of citizen science in addressing those gaps. So after the completion of our threat status of plant species during the period of 2003 to 2009, a lot of our species have been highlighted to have a lack of information, be it of abundance, of distribution, ecology, and threats. And so the red list assessments that happened at that period was not as comprehensive as it currently is. So SANBI, together with the Botanical Society, partnered to create a dedicated citizen science initiative, the Custodians of Rare and Endangered Wildflowers. It's a network of just over 1,000 volunteers across the country, strategically recruited in areas of high plant endemism and threat. So this flagship plant conservation program generates accurate spatial data and population trend data to advise on our red list assessments. Further to that, we have the iNaturalist platform. And I really hope that you all are iNatters, and if not, you would soon become an iNatter. iNaturalist coordinates the Southern African, SANBI coordinates the Southern African iNaturalist community to fill biodiversity data gaps. And we prioritize taxonomic groups while expanding our citizen science network. So currently a huge focus is on inverts, and we started up smaller projects whereby we're collecting data on specific groups that our red list scientists are working on. So you can see the stats, and this is just a few weeks ago. We host the Great Southern BioBlitz and the City Nature Challenge, which are international events that happens once a year. It's a 4-day period. It's a challenge to get people interested in looking at biodiversity, in exploring and collecting data across various species. And iNaturalist has become quite a large platform in the country. In fact, we've actually surpassed the amount of collections that we have in South Africa's herbaria across the country. And you can see the numbers speak for itself. So we've far surpassed specimens, and lots more data is being collected. You can see it's over 3 million observations, lots of species. Yes, there's lots of abnormalities on iNat in terms of identification, and we're trying to get more and more taxonomists involved. Our sensitive species are blocked, and there's various other things happening in the background. So I'd really encourage you all to start collecting your data on iNat. So looking at where our plant taxa of conservation concern are, you can see the numbers, well, the percentages is the amount of data that crew volunteers have collected over the past 2 decades. And you can also then see how much of taxa that we still need to monitor. So although we've been going for 2 decades and we've done a remarkable amount of work, there's still a lot more work that needs to happen in the plant conservation sector. Again, if we look at our TOCC, so TOCC is taxa of conservation concern. They are those categories that are threatened, which is critically endangered, and vulnerable, as well as other categories like rare, near threatened, declining, critically rare. So you can see the table has all of the numbers of, showing the red list, the species in the different red list categories, as well as then the iNat records in green. And you can see that we've been focusing a lot on collecting data on the threatened categories first. So over the past year, crews conducted over 500 field trips in just one field season. And if we think about the number of times that we managed to get onto the field, being paid employees or students, this is quite remarkable to have that amount of vast data coming through every field season. Another major thing that we do is species recovery and restoration. So we already know that our species are at a brink of extinction. How are we now combating extinction? How are we helping the species out there in the wild? And so we've just last year, together with our red list scientists and various restoration experts in the country, began doing a ranking system for our species of conservation concern to look at which species are a high priority of recovery. So you can see again the stats, just under 300 plant taxa in priority 1, which means if we don't save these species within the next 5 to 10 years, they're going to be extinct. And so we've come up with this major tree. So this decision tree is just part of the story, but just to show you just how much of detail we go into when we're looking at which species require recovery and what type of recovery is required. And so you can see there's interventions that we require, there's what data do we have for those particular plant groups, which stakeholders are going to be able to assist, and what sort of interventions are needed. So sometimes at a site, we just need to clear invasive aliens. Sometimes we need that site to be protected under biodiversity stewardship or maybe under protected area management via the provinces. So there's various interventions that needs to be employed. And so just to round off, citizen science data really informing decision-making. This is a SANBI value chain, and I know it's a lot of information, but if you just think of it as a 3-story building, and at the forefront is what we do. Across all of the work that we do is building capacity in the sector. At the bottom is our foundations of biodiversity, and there's where we're collecting our data. So our volunteers are out there collecting threat data out there in the field. We collect, we analyze the data using the red list, and we provide that to the red list, to the volunteers. And then at the second row, we are building that data. So now that we've got the data, we do our assessments, and so we can rank the extinction rate of our species. And then that science into policy, we're then informing the NBA and various other things, as I've mentioned at the beginning, that we're currently working on the next National Biodiversity Assessment Report. And all of this works together with the provinces and municipalities. So our citizen science data feeds into all levels of government and decision-making. And just some concluding remarks, so I'm just going to whiz through these. Just to emphasize how revolutionary citizen science is, not just in the plant conservation sector, but also in other taxonomic groups. We're finding that we're really bridging the gap between the researchers and the public, and we need more strong partnerships across the board, be it our stakeholders in academia and in government, as well as in the private sector, to collaborate and to drive successful conservation efforts. Oopsie. With that, I thank you.
Thank you, Suvarna. We've got some time for some questions. Have we got a quick question for Suvarna? Okay, if not, great, we can save a bit of time. Okay, so our next speaker is going to be Helga Knoetze. She's from Sion, the arid node, and she'll be telling us a bit about elephant megacarcasses and how they drive local plant species patterns.
Thank you, Cindy. Good afternoon, everyone. And yes, it's such a privilege to come and share some caucus results with you on the 60th annual Congress for the GSSA. So results that I'm going to present today form part of a larger study where we are looking at the ecological legacy effects of megacarcasses in African savanna ecosystems. And as you can see, this is a multidisciplinary study, and we are a bunch of people looking at in and around these carcasses, of course, at vegetation dynamics, soil microbes, large herbivores, scavengers, bone fields, and even the behavior of other elephants. So we know that decomposing large vertebrate carcasses form an inherent part of natural ecosystems. They are natural disturbance. They are a localized source of nutrient enrichment, and they have strong impacts on ecosystem biogeochemistry and also the community dynamics of vascular plants, large herbivores, scavengers, soil microbes, and invertebrates. And although we have a clear understanding of what happens in marine ecosystems where the megacarcasses of whales create these oases of biodiversity and productivity that can last for many years, we know we have limited knowledge on individual carcass effects on the spatiotemporal heterogeneity in terrestrial ecosystem processes. And this is specifically true and surprisingly true for the largest land animal, or the African elephant. We know little about how the nutrients from these megacarcasses affect savanna ecosystems. And given that elephant mortality is a natural part of the savanna, and annual mortality, for example, in the Kruger Park can total several hundred individuals per year. And the nutrient pulses of about 800 kilograms of carbon, 125 kilograms of phosphorus, and 300 kilograms of nitrogen for a single 4-ton individual. And the impacts of these nutrient pulses are likely to operate at a landscape scale. Now, although we still have a lot to learn about megacarcasses, through our ongoing research in the Kruger National Park, we have started to fill this gap a little bit. So we know now that elephant megacarcasses do increase local nutrient pools in savanna soil and plants. And we also know that these carcass-derived nutrients underpin floristically and structurally distinct plant communities. So yes, megacarcass sites are considered nutrient hotspots. They support mainly palatable species. So therefore, they do play an important ecological role in terrestrial ecosystems of semi-arid savannas. They influence herbaceous plant community composition and structure, and there's increasing emerging evidence that they do have long-lasting legacy effects on the environment. So therefore, we anticipated that elephant megacarcasses will also drive local patterns in plant diversity and richness. So here we explored richness and diversity patterns of plant communities that we sampled across megacarcasses of different ages, and we sampled these carcasses on granitic and basaltic soil in the Kruger National Park. And for the purpose of this talk, I'll be focusing on species richness, Shannon and Simpson's diversity, and species evenness. And to look at these different diversity indices, we established 4 transects of 15 meters each in the 4 cardinal directions at each of the carcasses. And we sampled vegetation within 1 meter squared subplots, which we placed at various distances away from the carcass. As you can see there at 1 meter, 2.5, 5, 10, and 14 meters away from the megacarcasses. And within these subplots, we identified all life forms, and we also noted percentage cover of vegetation, bare soil, bone, rock, and litter and dung. So what did we find? The CCA told us that soil type is the main driver of community composition. And we can clearly see that basaltic plots and granitic plots separated along axis 1, and that explained approximately 24% of the variation. Distance correlated strongly with the second axis, which explained an additional about 14%, and the age showed a negative association with both of these axes. However, plots sampled on basaltic soil correlated more strongly with older carcass sites. So I quickly went to have a look, and the carcasses that we sampled on basaltic soil had an average age of 3 years old, whilst carcasses sampled on granitic soil had an average age of 1 year old. So at first I wanted to have a coarser look, so I didn't irrespective of soil type, I just had a look at distance away from the carcass. So irrespective of soil type, I just wanted to see what the patterns are that will be emerging. And we found significantly lower richness and diversity closest to the megacarcasses up until 1 meter away. And we found a general trend of increasing richness and diversity with increasing distance away from the megacarcass. Now, referring back to the CCA, I wanted to see if the older carcass sites, which was typically the basaltic sites, revealed similar patterns in terms of richness and diversity. And sure enough, they also revealed significantly lower levels of richness and diversity closest to the megacarcass site. And again, a similar pattern of increasing species richness and diversity with increasing distance away from the carcass. So now I was interested in what was happening at the fresher carcass sites, and it was interesting, or maybe anticipated, that the zone of impact at the fresher carcass sites were a bit bigger. So we could see up until 3.5 meters, richness and diversity was significantly lower. So as we would expect, the fresher carcass still has a bigger effect a bit further away from the carcass as well. But similarly to basaltic soil, a general trend of increasing richness and diversity with increasing distance away from the carcass. So how can we explain these patterns? So we definitely saw suppressed diversity and richness closer to the carcass sites. And this could be attributed to extreme nutrient pulses from the elephant megacarcasses. And these extreme pulses create toxic soil. And then this prevents plants from growing. And this is specifically true for high concentrations of ammonium, which we also found in the younger or the fresher carcass sites. We did find toxic levels of ammonium. And then a second possibility could be due to the increased trampling and soil compaction close to the megacarcasses, that only certain species adapted for such conditions were able to persist there. Then in terms of species evenness, we found, conversely to richness and diversity, that species evenness was significantly the highest, closest to the megacarcass sites. And this could possibly be due to the dominance of certain species adapted to elevated soil nutrient levels and increased trampling. And for those of you who were here last year or not here in Harib, I did talk about at the carcass site, we found mainly palatable species and species with a prostrate growth form, like Cindy explained earlier, growing flat towards the soil surface, which is an adaptation for increased trampling and being utilized. And then interestingly, this was specifically true for Hluhluwe-Imfolozi-Park. This was a dominant species that we actually found at most of the carcass sites. We also found elevated foliar nitrogen levels in the species. And then we also found that this species seems to have a higher degree of ammonium tolerance because we really found them at the fresher sites as well. So elephant megacarcasses do drive local patterns in plant diversity and richness across basaltic and granitic semi-arid ecosystems. The extreme nutrient pulses from these megacarcasses cause unfavorable or toxic conditions for plant growth, leading to suppressed richness and diversity. And these effects are, of course, stronger or more observable in the fresher carcass sites. Elephant megacarcasses have long-term legacy effects on richness and diversity, as we saw. Even on an average of 3 years old, the carcasses still show large effects of these megacarcasses with significantly lower species richness. So elephant megacarcasses represent a relatively unexplored and unique disturbance type in savanna ecosystems. Their effects on vegetation species and diversity patterns that we demonstrate here adds to the spatial complexity of those semi-arid ecosystems in which they still occur. And then the widespread loss of megaherbivores and their carcasses across their native ranges reflect the loss of such an important driver of community dynamics and landscape heterogeneity. And with that, I would like to thank you very much.
Thank you, Helga. I must say, I'm very excited to see that you measured individual FOB responses. Okay, so our next speaker is going to be Debbie Jewitt. She's from KZN Wildlife, and she'll be telling us about hungry caterpillars.
So once upon a time, there was a caterpillar called lienardi, and his other brothers lienardi, and his other cousins lienardi. Now, lienardi lived in Hluhluwe-Imfolozi-Park, but his cousins can occur in sub-Saharan Africa, including Madagascar, the Comores, and Mauritius. Now, lienardi is a semi-looper caterpillar, and he likes to eat a lot of different plants, 44 known species of plants. He goes through 6 larval end stages before the final one reaches about 70 millimeters in length. And then they pupate, and they just pupate in the leaf debris, or they burrow shallowly into the soil, and they make a soil encrusted cocoon. Now, the variation depends a lot. There's a lot of variation in the larval color, as well as in the adult, in the forewings. So it's quite hard to identify them. And one looks at the hind wing pattern in order to identify the moth itself. And these moths can migrate, and they fly at night, and they lay a single egg. Now, lienardi can get into trouble with farmers because he likes to eat the leaves of eucalypts and wattles and castor oil plants, but then he likes also eating the fruits like of citrus plants, mangoes, apples, apricots, peaches, prickly pears. But the bad name he gets is actually unjustified because lienardi's proboscis is not a piercing type, so the fruit has to be damaged already before he can eat that. So he's a secondary feeder, not a primary feeder. And this was confirmed by electron micrographs. So in March of this year, they were really, really hungry. This was in Hluhluwe-Imfolozi-Park, and we can see that extensive areas of vegetation were defoliated, and these happened to be all Tambuti vegetation associations. I then looked at the same area under satellite imagery, and I could very easily map out using a false color composite the Tambuti vegetation compositions. So those are the dark red areas, and I've just drawn lines around in yellow. So now we can go back in time, and we can see that on the 8th of February, it wasn't really visible, the defoliation of these vegetation associations. But by the 5th of March, this was clearly visible on the satellite imagery, and extensive areas were defoliated, spreading into the neighboring vegetation. But then by the 17th of April, the vegetation had largely recovered. So I was wondering, what are the conditions that favor the outbreak of lienardi and his friends? So looking at precipitation and temperature variables for the last 5 years, we can see that in HRP, it is a summer rainfall area and a dry winter, and temperatures range between 10 degrees minimum and 30 degrees maximum on average. If we look more closely at what was happening to precipitation, we can see that in the June preceding the outbreak, we had a lot of rain in June in a dry period. It was 74 mils of rain compared to an average of less than 20 mils in non-outbreak years. Now this helps the pupae to survive. They don't desiccate over that period. But then we had really good spring rains in September, 149 mils, which was 4 to 5 times the amount of spring rain in other non-outbreak years. So this helps the vegetation to get a good flush and to get going. But then we had a dry early summer period in November, and this was significantly lower than in the preceding years, other than in November of 2021. So this helps. It reduces the flush of the vegetation, and it also depresses the predatory species of insects that might prey on lienardi. And then it synchronizes the flush of vegetation better to lienardi's life cycle. So now we can have a look at the summer rainfall. So rainfall was very good in January and February of the outbreak year. But what's really important here is that it was light to moderate rainfall and near continuous. So in January of that year, we had 19 days of rain, and in February, it was 24 out of 28 days had rainfall. This pushes up your humidity levels quite nicely. But wait, I can hear you say, other years had higher rainfall. Yes, but the one was due to Cyclone Eloise, and then the second one was due to a La Nina event and a cutoff low. So what this does is cause flooding. And there was extensive damage within the park and outside. And in fact, a state of disaster was declared. So what happens is that these little eggs and the caterpillars get washed away. It's not only your bridges that get washed away in these extreme events. So now if we have a look at temperatures, we can see that not only was the preceding winter wetter, it was also quite a lot warmer. And on average, it was 1 to 2 degrees warmer in winter than in the preceding 5 years. We can also see that the temperature was higher, but remember, we had lots of cloudy weather. So it wasn't this hot, dry weather. It was nice and humid and warm. So on average, 1 to 2 degrees warmer than the preceding years. And more importantly, the night temperature was much warmer, 1 to 3 degrees warmer than the preceding years. Now, why is this important? The moth flies at night. Warm temperatures boost metabolic activity, allows the moths to fly and to lay their eggs. And the caterpillars don't dry out, they've got little soft bodies. So in summary then, multiple climatic factors acted in concert to create ideal conditions for lienardi's survival. And it was a combination and timing of factors rather than any single extreme factor that made the 25 outbreak so severe. So this was your mild and wettish winter, promoting your larval survival. We had a spring flush and then a synchronization of the main flush with the insect life cycle in late summer. We had an early dry period that suppressed predator insect populations, and then more importantly, near continuous light to moderate late summer rainfall with warm nights and days. So the outbreak of lienardi coincided with an extensive armyworm infestation in other summer rainfall areas in South Africa. And this armyworm outbreak was said to be the worst on record. So the reasons that have been investigated for armyworm outbreak was an early summer rainfall, followed by midsummer dry spell and significant rainfall from January onwards, combined with warm temperatures similar to lienardi's. So how does one go about controlling lienardi? Well, in natural systems, no control is necessary. And as we've seen, the vegetation recovers very easily. And the control of the larvae is actually really impractical given that he eats so many different types of plants. So one wants to encourage your natural predators. However, in an orchard, and given that they're fruit-sucking moths, you still don't need to take any action against them because they're the secondary feeders. So what you could do then is to use varieties that produce crops outside of the major larval period, or you could have a variety that allows you to harvest your crops early. And then in commercial plantations, you would have an integrated pest management system, but limit the use of pesticides. So the natural predators include multiple different species of birds, and then parasitic flies, stink bugs, and wasps. So this was a short-term correlative study, and future research should really include monitoring and the development of a system dynamic model to better predict outbreaks and the population dynamics of the species. And then according to reviewer 2, I should have long-term climatic modeling of more than 20 years, population genetics, and predator exclusion experiments. So from lienardi and myself, thank you and goodbye. See you next year.
Thank you, Debbie. That was great. Has anyone got a burning question for lienardi and his friends? Yes, Richard.
That's a good question, and it's the first time it's been recorded. Well, I'm sure they've eaten it many times before, but in the literature, it was never recorded on Tamboti, and there's another species of Achaea that's more normally associated with eating Tamboti, and there are records of that in the Kruger, and people have reported it in Kruger, but this is an actual first record of Tamboti, and it's interesting because they just flattened the Tambotis. There was nothing there. It is, and yeah, these guys didn't seem to be bothered by it at all.
Thank you, Debbie. Okay. Our next speaker is Hazel Shirinda, and she's going to be telling us about land cover change patterns. She's from University of Pretoria.
Thank you very much. As mentioned, my name is Hazel, and I will be taking you to the City of Tshwane to look at the patterns and drivers of land cover change. So this study is part of a bigger project that is funded by the Danida Fellowship Center. To begin, across the globe, cities are continuing to expand and change at very big rate. And among the biggest drivers of these changes is the ever-increasing population and urban expansion and infrastructural development. Changes in land cover not only alters the city's original landscape, but it also leads to the loss of important ecosystems within urban landscapes. For this study, we particularly hone in on 2 ecologically sensitive areas within the City of Tshwane, areas of natural vegetation and riverine areas. Natural vegetation is important. It supports biodiversity. It helps in improving air quality. It also helps with regulating the microclimate in the city, acting as a carbon sink, improving the quality of life of the urban residents, and also providing important recreational areas. In addition to supporting biodiversity, riverine areas, on the other hand, also act as biodiversity corridors. They also help with flood prevention during seasons of heavy rainfall. And lastly, they also supply water again to the urban residents. So what happens when we lose these important ecological systems? Well, first of all, our habitats become fragmented. Again, we lose biodiversity at an accelerated rate. Increased flooding risks, which puts the urban residents in jeopardy. We also have poor air and water quality, which is a big problem, again, for the urban residents like myself. And then there's also the increased risk of the urban heat and island effect. Very big bad news for those who know about climate change. The City of Tshwane, it is one of the largest metros in the country, covering about 6,300 square kilometers and housing approximately 3.5 million people, myself included. It is in the grassland and savanna biome. And it is very diverse geographically, ecologically. It is home to many different settlements. You go from the wealthy parts to the poorest areas and informal settlements all over the city. And this now brings us to the aim of the study, which is to assess the patterns of and drivers of this land cover transformation in the city. The first objective of the study is to characterize land cover changes, followed by looking at what is driving these changes in the city. And the third objective looks at what is driving these changes, particularly in areas of natural vegetation. And lastly, what is driving these changes in riverine areas. To achieve that, we obtained maps from the South African National Land Cover Database, after which we quantified the changes for the year 1990 and 2022. If you don't know where the City of Tshwane is, somewhere there. After that, we ran GLMs to look at what is causing, what is driving this, the change in the whole city, in areas of natural vegetation and also in riverine areas. And this is just a list of the predictors that we used. Lastly, to capture the spatial heterogeneity in these changes, we ran a geographically weighted regression model for the whole city. So what did we find? Well, first, for how much change or how has land cover changed in the whole city over the 32-year period. First of all, we found that there is change across the city. Most of it is concentrated there in the northwest. However, it doesn't follow any particular direction. It is just randomly spread here and there. And between 1990 and 2022, over 25% of the landscape has been transformed. At first, I was like, that is very small. But looking at the area that we're working at and this period of 32 years, that is a very, very big problem. So here we have a comparison of what exactly has been happening in the city between 1990 and 2022. Here we see the map for 1990 and then the map for 2022. And these are our land covers. Already we can see that the green has been decreasing. So here by 2022, we have lost quite a significant portion of natural vegetation cover across the city. And we have gained a bit of cultivated land and residential areas. So in the 32-year period, we lost about 21% of our natural vegetation cover. However, we did gain something, cultivated land at about 96% and formal residences at about 38%. So what exactly is driving these changes that we're seeing? Well, here I show you what we had in 1990 and what we had in 2022. And if you look at this here, this is natural vegetation. Well, this part remained natural vegetation in 2022. However, significant portions of it was replaced by cultivated land or formal residences. So now to hone in on what is driving these changes, we looked at population density as one of our predictors, and we can see on that curve that population density and land cover change have a very lovely relationship. So population density is driving change across the city. Results from our geographically weighted regression shows that the relationship is positive throughout the city. However, it is weaker in some parts and stronger in some parts, like that part in the northwest. What about unemployment, the biggest problem in our country? Same story in the city of Tshwane. Unemployment is driving change at an alarming rate. And this is also supported by results from the geographically weighted model. So across the whole city, the change is positive. However, it is very strong in the city center. So that should paint a picture for you. However, when we look at distance to roads, we see that it has a negative relationship with change across the city. What this means is that closer to roads, we see a lot of change. So this can translate to accessibility, easier access, more development. However, looking across the city, this relationship is stronger in some parts of the city and weaker in other parts of the city. And in some other areas, it switches from negative to positive, which is interesting. Looking at the distance to rivers, we see the similar trend with distance to roads. In areas that are closer to roads, we see a lot of change. In areas that are closer to rivers, we see a lot of change. And looking at our geographically weighted regression, it is supported that yes, in areas closer to rivers, we experience a lot of change. However, in some parts of the city, this relationship is positive. In others, it is negative. As for the variation in elevation, the relationship is negative again across the city. So in areas that are low-lying, we see a lot of change, which makes sense. I wouldn't want to build a house on top of a hill. Looking at our results from the geographically weighted regression, this relationship is negative throughout the city. However, in the darker shades, this is where the change is most concentrated. And this is actually the city center. And lastly, we look at access to water. Again, it has a negative relationship. So areas that have limited access to water experience a lot of change than those that have more access to water. And the results from our geographically weighted regression kind of supports that. However, this relationship is also negative in some parts, but positive in some parts. Interesting stuff. So then we moved on to look at what drives change in areas of natural vegetation in the city of Tshwane. So this shows us natural vegetation in 1990 and 2022. Again, a very worrisome picture. We see that by 2022, most of the green has been replaced by red. So that indicates that most of the natural vegetation has been lost. To look at what is causing these losses or these changes in areas of natural vegetation, again, we see a similar trend with population density having a positive relationship with change in areas of natural vegetation in particular. Again, elevation variation has a negative relationship with change in these areas. Same story for distance to rivers and distance to roads. What this tells us is that areas of natural vegetation that are located closer to rivers and closer to roads tend to experience more change than areas that are far away from roads and rivers. And then also natural vegetation that is in low-lying areas tends to experience a lot of change. And then for our last objectives, what is driving change in riverine areas? Again, we see the similar thing with population density having a positive relationship with change. However, elevation variation, distance to roads and distance to rivers have a consistent negative relationship throughout. So now we have seen our results. What does this mean? Well, we have seen significant land cover changes in the city over the 32-year period, with major declines in areas of natural vegetation cover and major increases in cultivated land and population density. What this translates to is that population density, distance to roads, distance to rivers, these socioeconomic and proximity variables are driving change at an accelerated rate all across the city of Tshwane. And these are our major drivers of change. So again, distance to roads, distance to rivers, and elevation, our environmental variables, they have a very strong relationship. So this shows that low-lying areas are the most favorable for change. This could translate to terrain and also distance to roads. So accessibility translates to more development in the area. And then for distance to rivers, this could translate to maybe available land, easier access to water for farming. With unemployment, population density, and water access. So it means that if we have higher unemployment, higher population density, then we have a likelihood of seeing more change in such areas. So this change is not uniform across the city. One variable can have a positive effect on change in one part and have a negative effect on change on another part of the city. To conclude, this shift poses a serious threat to ecologically sensitive areas. This calls for sustainable urban planning, which must then balance with development with ecological conservation and preservation. And then in order for me to finish my PhD and get it on time, we also need to know and understand what exactly is driving land cover change across the city. This will also allow us to target and protect sensitive ecosystems. And that brings me to the end of my presentation. Thank you very much.
Thank you, Hazel. Unfortunately, we don't have enough time for the questions, but please do engage with Hazel during tea time. Now, our next 4 presenters are going to be doing short presentations. And the first one being Andri van Aardt, and he's going to be telling us something about range expansion. Oh, sorry, she.
Good afternoon, everyone. Today, I want to tell you more about the Karroid components present in the grassland. That's not my presentation. Oh, that's better. Okay. So today I want to tell you more about the Karroid components present in the grassland. So according to Messina and Rutherford, this vegetation type is an archipelago of isolated patches mostly restricted to dolerite outcrops. The dolerite result in shallow soils with a coarse texture leading to poor infiltration and high evaporation and runoff. The vegetation that we find there is mostly dwarf small-leaved Karroid succulent shrubs with some grasses occurring in the depressions and crevices. There's also an abundance of geophytic herbs and shrubs in areas where root penetration is possible. So now you might wonder why the interest in this vegetation type. So vegetation or urbanization, mining and agriculture pose a threat to this vegetation type, and very little thereof is conserved in the Free State's National Botanical Garden. Furthermore, there's also uncertainty about the protection status. SANBI sees it as vulnerable, while the Department of Environmental Affairs sees it as poorly protected. Lastly, there's very limited literature with regards to this vegetation type. So in 1937, Potts and Titmarsh described this vegetation type as unique islands of succulents that's dominated by karoo shrubs, while some surveys were also done in 2002 and 2003. These indicated strong affinity with vegetation from the arid west and strong grass components that can possibly be because of the microhabitat that is being controlled by the soil. So in the following maps, that is the extent of what Messina and Rutherford actually mapped this vegetation type as. However, in 2017, Prof. Johan de Pre refined the vegetation maps of the Free State because of the knowledge that he had. So he then indicated that there's the ones in green that he proposed that also should be part of this Karroid vegetation type. So we surveyed some of the mapped ones by Messina and Rutherford, as well as those ones proposed by Prof. de Pre, by doing over 200 sample plots in both the mapped and proposed sections. All of the species that are indicated in green here on the tables, so we did a classification to determine the communities, is actually indicative by Messina and Rutherford as being important species that are being used to identify this vegetation type. So I want to draw your attention to this last part of the tables, which is the species that grow throughout all of the communities found within this vegetation type. And most of them contain species that have been identified by Messina and Rutherford as being important. So in conclusion, the investigation of this vegetation type is still ongoing because it's much more extensive than what has been mapped before. But thus far, we've identified several communities that we can actually add on various topographical positions. So at the crest, we have communities that are dominated by succulents on bare rocks, or you can have a mixture of succulents and grasses on bare rocks. Or where there's deeper soil, you will have mostly grassland-dominated communities. And then as we move to the shoulder of these koppies, you will find vegetation that is a combination of grasses, ferns, and some succulents with scattered trees. There's also some of the trees. And then as we move to the plains, we find various grasses, succulents, and bulbs. We are currently still investigating some of the proposed sections, and we hope that our results will help in refining the plant communities of the Bloemfontein Karroid shrubland.
Thank you, Andri. Sorry, no time for questions, but please do engage with Andri during tea time. Our next presenter is Masibonge Gxasheka, and they will be telling us about landscape topography, soil acidity, how they drive floristic composition and diversity.
Good afternoon, ladies and gentlemen. My name is Masibonge, as already been introduced. Just waiting, okay. Yes, I'm here just to give you an overview on the landscape topography, as well as the soil acidity. Yes, since my presentation is too short, I won't be able to dwell much in some of the information, but what is key is to understand how topography is influencing the Euphorbundos that is affecting rangeland in the Eastern Cape. So this is one of the rangeland that is being affected by the species forming the monocultural stands everywhere it goes, and it replaces most of the areas. And our study was basically selected in Gala, as you can see, sorry. And we tried to look at the home distance sites, which is the first one is Tsengiwe, and the intermediate site was Apamle, and the far site was Manzimdaga. These sites, they were sitting at Tsomo Grassland. It's not it's not what you call a savanna grassland. We have to just understand that quickly. And in that area, in those sites, we selected sharp encroachment indigenous grasslands, and we make sure that we select the one that are facing north. And in each site, we divided the slope positions. And within each slope position, the 6 plots of 10 by 10 were selected, and we measured the vegetation structure within those. And we tried to also measure the herbaceous vegetation, and as well as the basal cover using levee bridge. The quadrants were 0.5 by 0.5. So samples were also collected just to try to look at how they influence the general vegetation. These are the soil parameters that we measured using the soil auger. These are the formulas that we use to analyze some of the species diversities, as well as calculating the cover. These are our statistics. Just to note that we did not combine the sites, we analyzed them separately. But what we found here is that the structure of the plant itself was shorter in the downslope, and as well as having higher density, but we check most of the time that this was happening in the far side, which is Manzimdaga, meaning that the disturbance could have override the influence of landscape position in the near and the medial side. When it comes to the herbaceous vegetation, we found only the grass richness that was higher in the downslope compared to upper slope position, again, in that particular side that is far. And this particular variable was more associated, according to our PCA, with the pH, which was more acidic. And we thought as well it could also link to some of other soil parameters that we did not measure, like soil moisture at the downslope position. Those areas were dominated mostly by Ulunus muticus across the slope position, as you can see there. And there's only one side that was having a little bit of variation in terms of the species composition. But generally, it shows that there is something that is happening within those ecosystem. I superlink this slide to explain that. I just want to show you how as the pH was linked with the species richness. We concluded that landscape position indeed does influence the structural growth of that particular species, meaning that the attention has to be given in terms of management. And the issue of the species richness, there's a need of management that also needs to be involved in trying to improve those areas. And soil acidity, yes, we acknowledge and we give perspective in terms of the microclimate conditions as GIS that needs to be integrated so that we can understand the distribution. And stakeholders in the Eastern Cape, they must start to work on the species before we start approaching other provinces, the neighboring provinces, particularly Free State and Western Cape. Thank you.
Thank you, Masibonge. Our next presenter is going to be Thandwayo Mbambo. She's currently doing a PhD at UKZN, and she will tell us about land use effects on forb diversity and life history traits.
Good afternoon, everybody. My name is Thandwayo, and this is my project proposal. I am wanting to understand how do land use effects on forb diversity and life history trait distributions in montane grasslands. So montane grasslands are known for their high biodiversity and endemisms, and these systems are shaped by fire, grazing, together with shallow soils that prevent tree growth. They also offer key ecosystem services like nutrient cycling, soil retention, carbon storage, biodiversity, and water regulation. However, these systems are threatened by climate change, woody encroachment, and habitat modifications. Forbs are an important part of these ecosystems because they are huge contributors to their biodiversity and ecosystem functioning, as well as maintaining ecosystem resilience in these ecosystems. There is not enough data compared to grasses with Forbes, especially looking at seedling recruitment and life history traits. And these are important to understand how these respond to disturbances. And also, because Forbes are much less than grasses, they are also part of the red list. So my research questions are, how does historical afforestation affect plant diversity and productivity? How do Forbes life history strategies and recruitment differ between primary and secondary grasslands? How do fire and grazing and time-sense disturbance affect species composition? And how do microclimate and soil properties affect Forbes and seedling establishment? So to answer the first question, I'm going to do a systematic review looking at how historical land uses have affected biodiversity and productivity in grasslands, and then to look at how primary and secondary grasslands differ in terms of richness. I'm going to do Forbes compositions and also do key traits between these two grasslands. The key traits I'm going to look at are SLA, dispersal mode, number of seeds per plant. And then to monitor seedling recruitment, I'm going to do seedling counts and identify those. And then to look at how microclimate affects seedling establishment, I'm going to use TMS loggers. They measure soil temperature and moisture. And then to look at the soil properties, I will measure soil nutrients, texture, bulk density, and rock cover. Okay, so why does the study matter? It matters because Forbes are important because of their biodiversity, but they are often overlooked in grassland research. And the legacy effects of land use like afforestation and grazing can obscure present-day vegetation patterns. And understanding how all these three things like disturbance and environment and history interact can help us improve restoration success, support climate resilient management, and guide conservation strategies that go beyond grass biomass. Thank you.
We've got some time for a quick question. Yes, Susie.
So important.
I'll consider that, thanks.
Okay, that was quick. Thank you, Tsandrayo. We look forward to seeing the preliminary results next year. Our last speaker for this session is Helga van der Merwe, and she is from semi-arid node. She'll be telling us about long-term population trends.
Thank you very much. My project is on the long-term population trends of an endemic dwarf succulent in the succulent Karoo, and I'd like to thank my co-author Sue Milton, who put in a lot of time and effort into this project. The Prince Albert Vygie is an endangered endemic dwarf succulent, and it's confined to the quartzite pavements. It's an edaphic specialist. It's vulnerable to physical disturbance, increased temperatures, and then changes in amount and timing of rainfall under climate change. The aim of our project was to track this population change in this dwarf succulent over 20 years under natural conditions. Various populations were surveyed at various elevation levels. They were surveyed in 2002 following above average rainfall, and then in 2020 and 2021 during a prolonged drought, and then again in 2023 after drought-breaking rains. An additional site was also sampled in 2023 for the first time. The populations were delineated by means of polygons with edge plots and center plots. Various parameters were measured within these plots, such as abundance, plant size, the microsite, was it in an open or a shaded microsite, and capsule numbers and flowers. The temperature and humidity at the soil surface were measured with portable data loggers and weather sourced from SAWS and SAON. The weather data confirmed the prolonged drought and the high maximum temperatures exceeding 40 degrees. The beginning of the drought was 2015. Before that, there were less than 5 days per year with temperatures exceeding 40 degrees. During the drought, there were between 5 and 10 days a year exceeding 40 degrees. There were no differences between the edge and the center plots. The shaded microsites were significantly cooler and they stayed moist for longer after a rainfall event. The plants in the shaded microsites were also larger. The populations all declined. However, there were significant differences between the sites and there was a decrease in mortality with increased elevation. The highest mortality was in the smallest individuals. Population structure was found to be influenced by grazing intensity, the microsites, whether open or shaded, and the initial population size. Our findings support the predictions of warming or drought and warming having a negative effect on endemic dwarf succulents. The elevation seems to have provided a refuge for heat-induced mortality, and therefore elevated sites should be considered for protection. Shade also reduced heat stress and evapotranspiration, highlighting the importance of grazing management, especially heavy grazing, which reduces shrub cover and shading of the individuals, but also causes direct damage to the plants. Thank you.
Thank you, Helga. We've got time for a quick question or comment. Okay, if not, great. That takes us to an end of our session. Can we give a round of applause for all our speakers? Thank you. Please enjoy tea.